In our initial lectures, we discussed the thyroid gland in detail. Now, moving one step further, let us shift our attention to another small but extremely important endocrine gland, the parathyroid gland. Although these glands are closely associated with the thyroid gland, they have a completely different function, primarily regulating calcium and phosphate balance in the body. Normally, there are 4 parathyroid glands located on the posterior surface of the thyroid gland, with one gland situated behind each upper and lower pole. Here we can see the thyroid gland. Now, if we rotate the model and look at its posterior or back surface and temporarily remove the overlying structures for a clearer view, you can easily appreciate these 4 tiny glands located on the back of the thyroid gland. These are the parathyroid glands. Typically, there are 4 parathyroid glands, 2 superior and 2 inferior. Each parathyroid gland is about 6 millimeters long, 3 millimeters wide, and 2 millimeters thick, and has a macroscopic appearance of dark brown fat. The parathyroid glands are difficult to locate during thyroid operations because they often look like just another lobule of the thyroid gland. For this reason, total or subtotal thyroidectomy frequently resulted in removal of the parathyroid glands as well. Interestingly, the body can function quite well even if some parathyroid tissue is lost. For example, removal of half of the glands usually causes no significant problems. And even if only a small amount of parathyroid tissue remains, it can gradually enlarge and compensate for the lost glands. Furthermore, when we look at the microscopic structure of the parathyroid gland, we find that it is composed mainly of chief cells, along with a smaller number of oxyphil cells. The chief cells are particularly important because they produce and secrete parathyroid hormone, or PTH, which is the principal hormone responsible for maintaining calcium homeostasis. On the other hand, the exact function of oxyphil cells remains uncertain, although they are generally believed to be inactive or modified chief cells that have lost most of their hormone secreting ability. Now, after the anatomy and cellular composition of the parathyroid glands, the next important question is, what exactly does parathyroid hormone, or PTH, do in the body? To answer this, let's consider what happens when PTH is suddenly introduced into the bloodstream. When parathyroid hormone is suddenly infused into an animal and the infusion is continued for several hours, characteristic changes occur in the blood levels of calcium and phosphate. Initially, the calcium concentration in the extracellular fluid begins to rise gradually, and within about 4 hours, it reaches a plateau. Meanwhile, phosphate levels behave quite differently. Instead of increasing, they fall rapidly and reach a low level within just 1 to 2 hours. This rise in blood calcium occurs mainly because PTH acts on the bones, where it stimulates the release of calcium into the bloodstream. In addition, PTH also acts on the kidneys, promoting calcium reabsorption and thereby reducing its loss in urine. As a result, the concentration of calcium in the extracellular fluid gradually increases. In contrast, although PTH acts on the bones and releases phosphate into the bloodstream along with calcium, its effect on the kidneys is much stronger. At the level of the kidneys, PTH markedly increases phosphate excretion in the urine. Consequently, this renal loss of phosphate outweighs the phosphate entering the blood from bone, leading to a rapid decline in blood phosphate concentration. Thus, overall, PTH increases extracellular calcium levels while simultaneously decreasing extracellular phosphate levels. Now, we have seen that one of the major actions of PTH is to mobilize calcium and phosphate from bone. The next logical question is, what is the mechanism behind this process? How does PTH actually cause these minerals to move from bone into the bloodstream? To answer this, we must first understand the different types of cells present in bone and then explore the two phases through which PTH exerts its effects, a rapid phase and a slow phase. Before discussing the rapid phase of PTH action, let's first review the major cells present in bone. First, we have osteoblasts, which are bone-forming cells located mainly on the surface of bone. Their primary function is to synthesize new bone matrix and promote bone mineralization. Their main function of osteoblast is to produce a substance called osteoid, which is the organic part of the bone matrix. By organic, we mean the living, protein-rich portion of bone that is made mainly of type 1 collagen fibers and other proteins. At this stage, the osteoid is still soft and flexible because it has not yet been mineralized. Its main role is to provide the basic shape and organized framework of the future bone, allowing calcium and phosphate to be deposited in the correct arrangement. After the osteoid framework has been laid down, calcium and phosphate ions are deposited onto it as hydroxyapatite crystals. This process is called mineralization. These crystals become attached to the collagen framework and gradually harden it. To understand this, think about building a house. First, workers create a framework using steel rods. Then, cement is poured onto that framework. If you poured cement without any framework, the structure would not be properly organized or strong. Similarly, osteoblasts first lay down a soft osteoid framework, and later, calcium and phosphate are deposited into it to make the bone hard and strong. After the osteoid has been mineralized and new bone has formed, during this process, some osteoblasts become trapped within the newly formed bone. Over time, these trapped cells undergo a change and become osteocytes, essentially mature osteoblasts that are now embedded within the bone matrix. In simple terms, an osteocyte is a mature osteoblast that is enclosed within the bone matrix. Once this transformation occurs, the cell's role changes from building new bone to maintaining and monitoring the health of the existing bone tissue. These osteocytes are the most abundant cells in bone and are interconnected with one another through long cellular processes, forming an extensive communication network throughout the entire bone. Finally, we have osteoclasts, which are large multinucleated cells responsible for bone resorption. These cells break down bone and release calcium and phosphate into the bloodstream. Now, let's come back to the rapid phase of calcium and phosphate mobilization. If blood calcium starts to rise within minutes after PTH is released, osteoclasts cannot be responsible for this effect because the formation and activation of new osteoclasts takes several days. So the question is, how can PTH increase blood calcium so quickly? The answer lies in the osteoblasts and osteocytes. Although these cells are traditionally known for their role in bone formation, they also participate in the rapid mobilization of calcium from bone. Osteocytes are located within small cavities or spaces in the bone matrix called lacunae. In other words, a lacuna is simply the space that houses an osteocyte. Between the osteocyte and the surrounding wall of the lacuna is a thin layer of fluid known as bone fluid. As you can see in this image, osteocytes also possess long, thin cellular extensions that pass through tiny channels called canaliculi. These cellular extensions connect osteocytes with neighboring osteocytes and also with osteoblasts on the bone surface, forming an extensive communication network throughout the bone. The bone fluid lies in close contact with the mineralized bone, which is rich in calcium and phosphate. Because of this close relationship, small amounts of these minerals continuously move between the bone mineral and the bone fluid, maintaining a relatively high concentration of calcium within the bone fluid. Now, Let's see what happens when PTH is released. Both osteoblasts and osteocytes possess PTH receptors on their cell membranes. When PTH binds to these receptors, calcium is continuously transported from the bone fluid into osteocytes. Through their interconnected cellular extensions, which run within the canaliculi and connect osteocytes with one another and with osteoblasts on the bone surface, this calcium is transferred toward the bone surface. From there, it enters the extracellular fluid and eventually diffuses into nearby blood vessels, helping to maintain normal blood calcium levels. As a result, calcium is removed more rapidly from the bone fluid and transported into the extracellular fluid. Consequently, the calcium concentration within the bone fluid falls. To restore equilibrium, calcium phosphate salts stored in the nearby bone crystals begin dissolving and releasing calcium and phosphate into the bone fluid. This process continues as the calcium is pumped out into the extracellular fluid and ultimately enters the bloodstream. Therefore, blood calcium levels can rise within minutes of PTH secretion. Importantly, during this rapid phase, the actual collagen framework or organic matrix of bone is not destroyed. Only the mineral salts are mobilized from the bone. This rapid release of calcium and phosphate from bone without true bone desorption is known as osteolysis. Therefore, the rapid phase of PTH action is mediated primarily by osteocytes and osteoblasts. However, if PTH levels remain elevated for several days or longer, a second, much slower mechanism comes into play. Interestingly, PTH does not act directly on osteoclasts because these cells lack PTH receptors. Instead, PTH first activates osteoblasts, which then produce signaling molecules such as rank ligand and release macrophage colony stimulating factor. As illustrated in the figure, rank ligand and colony stimulating factor binds to receptors on pre-osteoclasts and stimulates their maturation into fully active osteoclasts. At the same time, PTH decreases the production of osteoprotegerin, a molecule that normally blocks osteoclast formation. As a result, the number and activity of osteoclasts increase significantly. These mature osteoclasts attach to the bone surface and begin resorbing bone by secreting acids and lysosomal enzymes, creating areas of bone desorption. Through this process, large amounts of calcium and phosphate are released into the bloodstream. Therefore, the rapid phase of PTH action involves osteocytes and osteoblasts releasing mineral from bone, whereas the slow phase involves osteoclast-mediated bone resorption. Although both mechanisms increase blood calcium levels, prolonged excess PTH can eventually lead to substantial bone loss and weakened bones because bone breakdown exceeds bone formation. So far, we have seen how PTH increases blood calcium levels by mobilizing calcium and phosphate from bone through both rapid and slow mechanisms. However, bone is not the only target organ of PTH. The kidneys also play a crucial role in maintaining calcium and phosphate balance. When PTH acts on the kidneys, it produces two important and opposite effects. First, it decreases the reabsorption of phosphate in the proximal tubules, causing more phosphate to be excreted in the urine. As a result, blood phosphate levels fall. At the same time, PTH increases the reabsorption of calcium, particularly in the thick ascending limb of the loop of Henle and the distal tubules. Consequently, less calcium is lost in the urine and more calcium is retained in the body, contributing to the rise in blood calcium levels. In addition, PTH also increases the reabsorption of magnesium and hydrogen ions, while reducing the reabsorption of sodium, potassium, and certain amino acids. Nevertheless, its most physiologically important renal actions are the conservation of calcium and the excretion of phosphate. In fact, this calcium conserving effect of PTH is essential for maintaining calcium homeostasis. Without PTH-induced calcium reabsorption, a significant amount of calcium would be continuously lost in the urine, which could eventually deplete calcium stores in both the extracellular fluid and the bones. Therefore, by promoting calcium retention while simultaneously enhancing phosphate excretion, the kidneys play a major role in mediating the overall effects of PTH on mineral balance. PTH also indirectly influences the intestines and thereby further contributes to maintaining normal mineral homeostasis. Interestingly, PTH does not act directly on the intestinal cells. Instead, it first acts on the kidneys, where it stimulates the conversion of vitamin D into its active form, 1,25-dihydroxycholecalciferol, also known as calcitriol. Consequently, the increased levels of calcitriol enhance the absorption of both calcium and phosphate from the intestine into the bloodstream. Therefore, in addition to mobilizing calcium from bone and conserving calcium in the kidneys, PTH also helps increase the entry of calcium and phosphate from the diet. Now that we have understood the actions of PTH on bone, kidneys, and the intestine, an important question arises. What regulates the secretion of PTH itself? The answer is the concentration of calcium ions in the extracellular fluid. In fact, calcium concentration is the single most important regulator of PTH secretion. Whenever the blood calcium level falls, even by a very small amount, the parathyroid glands respond within minutes by increasing the secretion of PTH. Furthermore, if this decrease in calcium persists for weeks or months, the parathyroid glands not only secrete more hormone but also enlarge in size, a process known as hypertrophy. For example, enlargement of the parathyroid glands can occur in conditions such as rickets, pregnancy, and lactation, where the body's demand for calcium is increased. Conversely, when blood calcium levels rise above normal, PTH secretion decreases, and over time, the parathyroid glands become less active and smaller in size. This can occur with a high calcium diet, excess vitamin D intake, or increased bone resorption due to causes other than PTH. But how do the parathyroid glands detect these changes in calcium levels? They do so through a specialized receptor on the surface of parathyroid cells known as the calcium sensing receptor. When blood calcium levels are high, calcium binds to and activates this receptor. As a result, intracellular signaling pathways are triggered, ultimately suppressing the secretion of PTH. On the other hand, when blood calcium levels fall, activation of the calcium sensing receptor decreases, removing this inhibition and allowing PTH secretion to increase. Therefore, low calcium stimulates PTH release, whereas high calcium suppresses it. Now that we have discussed parathyroid hormone in detail, let's briefly examine another hormone involved in calcium homeostasis, that is, calcitonin. In many respects, calcitonin has effects opposite to those of PTH. While PTH increases blood calcium levels, while calcitonin acts to lower them. However, it is important to remember that in humans, calcitonin plays a much less significant role in calcium regulation than PTH. Calcitonin is produced by specialized cells of the thyroid gland known as parafollicular cells or C cells, which are located between the thyroid follicles. The secretion of calcitonin is stimulated primarily by an increase in blood calcium concentration. Therefore, unlike PTH, which is released when calcium levels fall, Calcitonin is released when calcium levels rise. Once secreted calcitonin acts mainly on bone. Initially, it suppresses the activity of osteoclasts, the cells responsible for bone resorption. Consequently, less calcium is released from bone into the bloodstream and more calcium remains deposited within the bone matrix. In addition, with prolonged exposure, calcitonin reduces the formation of new osteoclasts, further decreasing bone resorption. As a result, blood calcium levels tend to fall. Calcitonin also exerts minor effects on the kidneys and intestines, generally opposing the actions of PTH. However, these effects are relatively weak and are usually considered physiologically insignificant. Despite these actions, the overall influence of calcitonin on calcium homeostasis in adult humans is surprisingly small. One reason is that any decrease in blood calcium caused by calcitonin quickly stimulates PTH secretion, which largely counteracts the effect of calcitonin. Furthermore, bone remodeling occurs relatively slowly in adults, limiting the hormone's impact on blood calcium levels. In fact, even complete removal of the thyroid gland and therefore loss of calcitonin secretion does not significantly alter long-term blood calcium concentration. Nevertheless, calcitonin is more important in children where bone remodeling is very active, and in certain bone disorders such as Paget disease, where excessive osteoclastic activity makes the hormone's inhibitory effects more noticeable.